Hello, everyone, and welcome to phyloseminar.org. The current theme is evolutionary ecology, and this is the third talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Dr. Mariana Braga, a Brazilian biologist who moved to Sweden to do her PhD in ecology. She is interested in understanding how ecological interactions evolve using butterflies and their host plants as a model system. Since completing her PhD, she did a postdoc in the US to dive more into development of inference methods, and now she's back in Sweden and Finland, working on a new project aiming at combining evolution of species interactions with projections of species distribution. Welcome, Mariana, and thank you for participating. And I should say actually 2 things. I'm especially grateful for Mariana for helping co-organize this session of talks, and also that this is work that was rewarded via a Publishers Award in SysBio recently. So special treat for us today. All right, thanks, Mariana. Take it away.
Thanks, Eric. That's very nice. Thank you for organizing this and for inviting me. It's a pleasure to talk to give a talk to philoseminars. Yeah, thanks to everybody who's over there watching. And also thanks to Chris and Wade, who gave the first 2 talks in this evolutionary ecology series. If you haven't checked their talks yet, please, it's worth going and checking it out. All right, so my work is mainly focused on intimate ecological interactions, especially parasitisms. And there is a common pattern observed in many different host parasite systems. But first, a disclaimer. I consider butterflies to be parasites, at least while there are caterpillars, because they live in a host plant without which they don't survive. So regardless if we're talking about butterflies and their host plants, or bird endoparasites, or fish ectoparasites, we see the same pattern in the distribution of host range, which is the number of hosts used per species. So as you can see in these graphs, most species use one or a few hosts, but there's a long tail of more generalist species. And here I just need to say one taxon. Very rarely it's just one species of host. And this variation in the number and identity of hosts produces variation also in the networks built by these interactions. So here we have 4 examples of 4 different bipartite networks. And bipartite networks are networks with 2 groups. For example, plants and galling insects, or prey and predators. And here, these 4 examples, they're very different. So in A and B, we have disconnected networks. They're sort of fragmented. And C and D, on the other hand, are very connected networks, which means that all the nodes, so all the taxa in the network, are connected directly or indirectly to all the other nodes in the network. And I've always been curious about which evolutionary processes might generate these properties. So there are 2 main hypotheses of host-driven diversification, and both were inspired by butterfly plant model systems. So in a paper in 2018, we showed that these 2 scenarios produce different network structures. So in the left side, there's the variability scenario that comes from the oscillation hypothesis. And in that scenario, it's heritability and variation in host use that produces or promotes butterfly diversification. And we show that this scenario produces network nestedness, and that it's mainly the ancestral hosts that are producing this pattern. And on the right side, we have the radiation scenario, which comes from Ehrlich and Raven's Escape and Radiate. And in that scenario, diversification happens in bursts. They're followed by, bursts following the colonization of new hosts. And we show that this scenario produces, each one of these bursts produces a new module in the network, thus increasing network modularity. And it's mainly the recent hosts that are producing this pattern.
Let's see,
So
Mariana, are you going to be going through this? That went by a little bit fast, like the nested versus.
Well,
is this going to? Do I need to follow this for the rest? I mean, I think I kind of understand, but like, if this is going to be key for understanding the rest of the talk, I'd like to hear a little bit more about the.
No, this is more to show you that different evolutionary processes produce different networks, and yes, but it's not going to be the focus on how we couple these processes to the pattern. So that's okay if you.
Great. I mean, it makes sense in the broad outlines. Thanks.
Yeah. Okay, so we found evidence that both of these scenarios have contributed to butterfly diversification when we looked at the networks of two butterfly families. So by analyzing empirical and simulated networks, we proposed a unified explanation for how hosts can drive parasite diversity and how network would change in response. But to test our proposed ideas, we needed to reconstruct ancestral networks. And that's not a trivial task. So if we have a phylogenetic tree for our parasite clade, and we want to reconstruct the process of gaining and losing hosts through time, the first thing that we need to do is to define the character that we will reconstruct. And the data that we usually have available is a phylogeny for our parasite clade and a matrix telling us which parasites interact with which hosts. And of the model, the methods there were usually available and people used most often to do ancestral state reconstructions of species interactions. They were often of three kinds. First one is focusing on the main host of each parasite species. So each parasite can only interact with one host. And as you can see here, then we are ignoring a lot of data because it's very rare that each parasite in the clades that we are interested in only uses one host. Another approach is to do the analysis for your parasite clade and one host at a time. But then the problem is that we're assuming that the interactions between the parasite clade and each host evolves independently. And that's probably not true. And then a third way that people usually tackle those questions is by reconstructing how the number of hosts or host range changes over time. But then we don't know anything about which hosts these parasites are using. So we believe that the character that we should use is host repertoire, which includes both the number of hosts, so host range, but also the identity of the host used. So instead of using one binary or multi-state character, why not combine the information for all possible hosts in a vector? And then each element of this vector represents one host. As you can imagine, this makes the analysis much more complicated and much harder for our computers to handle. That's where Michael comes in. So thanks to his work on biogeographic inference, we could develop our model of host repertoire evolution, which is implemented in RevBase. So the first important feature of our model is that species can use multiple hosts at any given time. So the model can take in much more data than previous methods. We can also consider potential hosts, which are hosts that parasites could use in nature, but they don't. In butterflies, we know that larvae can feed on many more plants than females lay eggs on. So a potential host for butterflies is a plant that the larvae can eat, but they don't use in nature because females don't identify this plant as a host. And our model has up to 3 states and 4 transition rates. So the transition from 0 to 1 means that the parasite gained the ability to use that host. And then from 1 to 2 means that the parasite gained the actual usage of that host. And then we have similarly 2 loss rates. And if we ignore the potential host state, the model is reduced to 2 states, so host and non-host, and 2 rates, a gain and 1 loss rate. Then the last main feature of the model is that the phylogenetic proximity between hosts can facilitate host gain. If phylogenetic or any other pairwise distance between hosts matters for the host gain, it will be easier for the parasites to colonize a host that is similar to the hosts it already uses. And there's a parameter that controls how much easier this is.
Quick question.
Yeah.
So do I understand correctly that there's a single rate that gets you both from 0 to 1 and 1 to 2?
No, there are 2 separate rates.
2 separate rates, and then there's 2 separate loss rates as well.
Yes.
Okay, got it.
Yeah.
And there's nothing, if I understand correctly, there's no interaction between existing hosts and gain or loss of hosts. It's like host independent, right? So if I have 5 hosts, the rate of me gaining an additional host is the same as if I have 0.
Well, the number of hosts per se doesn't affect it, but the phylogenetic proximity between the hosts can do it. So if you use hosts that are very similar to the host that you're trying to gain, that may facilitate. So that rate would be higher than if you're trying to colonize a host that is distantly related to the host that you're using. But that importance, so there's this parameter beta that we infer that tells us if the phylogenetic distance matters or not in our data. So if that parameter is 0, then it's an independence model. Then each host evolves independently. And as that parameter increases, then it's easier and easier to colonize closely related hosts.
So just to clarify, the functional form of this is like you have some baseline. I'm just trying to think about how these all get combined into a formula. It's that there's like...
You have a base rate, and then you have a rate modifier.
Which is multiplicative or additive?
Yes, multiplicative.
Okay. And that has to do with the existing hosts that you have in their phylogenetic relation.
The existing host the host that the parasite's using at that moment.
Okay. This is totally cool. Thanks.
Yeah. Yeah, so for more details about the model, of course, there's the paper in systematic biology, but there's also a tutorial that goes through the whole analysis in the RAP-based website. All right, so getting into a model system, or sorry, a study case with Pieridae. Right, so we got data for 66 butterfly genera and 50 host plant families. And we used the two-state model, so no potential hosts, to reconstruct the evolution of the network between Pieridae butterflies and their host plants. So this is what the data looks like. We have the butterflies in the rows and the hosts in the columns. And then each colored square represents an interaction. And each color is showing a different module. A module is a group of butterflies and plants that interact more with each other than with the rest of the network. Right, so first results, posterior distribution of our parameter estimates. So in this case, we only had two states. So we have two transition rates, a host gain and a host loss. And then we have the overall rate of host repertoire evolution and the phylogenetic distance power, beta. And we ran two different variants of the model, but it doesn't really matter now. So we can only focus on this result that we inferred an average 148 events along the diversification of Pieridae, and with about half of gains, half of losses, and that's about 6 events per every 100 million years per branch. So this is a quite low rate of evolution. Pieridae are very conservative in their host use. And then for ancestral states, we did a first reconstruction that we called a traditional ancestral state reconstruction, where each internal node in the butterfly tree, for each internal node in the butterfly tree, we got samples from the MCMC. Then we calculated posterior probability for each host at each node. And then we only kept the interactions with more than 90% posterior probability. So again, this is the extant interactions. And on the bottom, we have the plant phylogeny. And on the left, the butterfly phylogeny with the reconstructed ancestral states. Each square in the internal nodes represents one plant family, and they are colored by the modules in the extant network. So all of host plants, only 6, these 6 in the legend, were used with more than 90% posterior probability. And we can see in this reconstruction the origin of the 4 main modules of the network, with Fabaceae being used by the ancestor of all Pieridae and also the basal clades. Then a host shift from Fabaceae to Caparaceae at about 17 million years ago. But this shift has been reconstructed before. So the most interesting thing here for us that was a novel result was the reconstruction of this host range expansion at the top of the phylogeny, the origin of Pieridae, where they colonized Brassicaceae and Tropaeolaceae. They're the red hosts. while also retaining capraceae, the blue module. And capraceae was their ancestral host. So this was pretty cool that we got this result. But the real new thing is the second approach that we used to reconstruct host repertoires for all extant lineages at different points in time. So for each time point, so first we chose arbitrary time points in the past. Then for each time point, we found the extant lineages, butterfly lineages. Then we got the samples from the MCMC. Then we calculated the posterior probability for each host at each branch. And then we combined all of the interactions at the same time point to make a summary network. And we included all the interactions with more than 50% posterior probability. So these are the summary networks we reconstructed from 80 million years ago in the top left to 10 million years ago in the top right, plus the extant network in the bottom right. So let's look at which one of them. Right, so for each time point, we show the network and the butterfly tree. The thickness of the edges of the networks are proportional to the posterior probability of that interaction. And for the nodes, circles are butterflies and squares are plants. And the colors still show modules, but now we identify the modules in each network and then match them across time points based on the host plants that were in those modules. So in the beginning of Pieridae diversification, Fabaceae was the most likely host, followed by Caparaceae. Then at 60 million years ago, we see that those two modules became distinct. They separated, with Fabaceae being used by basal Pierids and Caparaceae being used by Pieridae, which is a subfamily. And comparing the network at 60 and 50 million years ago, we can see that the blue module really increased due to the diversification of Pieridae butterflies. And then at 40 million years ago, Laurentaceae and Santalaceae are colonized by a host shift. And Brassicaceae is colonized by a host range expansion and the host shift. And because most of the butterflies that became part of module M3, the red modules, when they colonized Brassicaceae, they also kept Caparaceae in their host repertoire. Modules M3 and M2 are now connected. And at 20 million years ago, the network increased with butterfly diversification and colonization of new hosts. But recolonisations of ancestral hosts connected new modules to old modules. So now the network is almost all connected. And this continued through 10 million years ago and all the way to the present. All right, cool. Then you can ask me, okay, but how robust are these modules? Because it is really hard to calculate posterior probabilities for modules, we validated the modules of the summary networks by calculating how often each pair of nodes, so both butterflies and plants, were assigned to the same module. So we got 100 samples from the MCMC, found the networks at each time point, at each sample, identified the modules at each network, and then calculated how often two taxa were placed in the same modules for all the pairs of nodes. So each matrix here contains all the butterflies and plants present at each time point in the rows and the columns. And the more gray cells, the less robust the modules are, because the rows and columns are colored by the modules in the summary network, and then the gray or when nodes that were placed in different modules in the summary network were found in the same module in the sampled networks. So the more gray, the less robust the modules are. So we can see that it's only at 80 and 70 million years ago that the modules are not robust. And that's sort of expected. The further back we go in time, the more uncertainty we have. So it's more likely that there was only one module back then. But overall, we're pretty confident in our reconstruction. So the last thing we did was to look at how the structure of the network changed over time in terms of modularity and nestedness. If you remember, we talked about this in the beginning. So because now we are talking about one value for each network, now we could get a proper posterior distribution. So what I'm showing here is the distribution of z-scores, meaning how much more or less nested or modular each network is than expected by chance, given the size of the network. Because these indices correlate with network size, we need to standardize them before we can compare different networks. So for that, we calculated modularity and nestedness for 100 sample networks and for 100 null networks for each sampled network. Then we calculated the z-score. So each violin shows the distribution of z-scores for the sampled networks at each time point, and the line shows the average values. The orange dots mark the values that are higher than expected by chance, and the black dot shows the values for the present-day network. As you can see, both modularity and nestedness were first detectable 30 million years ago, and that means that the structure we see today in the extant network is an evolutionarily stable structure. So by combining our model of host repertoire evolution with network analysis, we could test ideas about the evolution of ecological networks. We provide a new probabilistic representation of ancestral interactions that makes fuller use of the posterior distribution of ancestral states. And by doing so, we could reconstruct specific host shifts, host range expansions, and recolonisations that have shaped the Pieridae angiosperm network. So in my GitHub, you will find all the analysis for all the code for the analysis of the paper in ecology letters in this repository called Pieridae host rep. And we also have your writing an R package that's almost ready. It's called EvoNets, and it's also available from GitHub, where you can do analysis of the output of the host repertoire reconstruction from RapBase. And there's all like the plotting functions and so on. So if you're interested, just go to my GitHub. So for future developments, we have several things that we want to follow, and there's not enough people or time for it. But so one big assumption that we make is that hosts don't evolve. So all hosts are older than the parasite clade. And to tackle this, we could then develop a model with the time stratified process, where at each like different time intervals, then we would have different increasing number of hosts available from the past to the present. The complications are that then, of course, the trait itself, the host repertoire, changes over time, and then the phylogenetic distance between hosts also changes. Yeah. Well, we also assume that the trees are known. So maybe in the future, we could include phylogenetic uncertainty. We also use the phylogenetic distance between hosts. And I don't remember if I mentioned this, but we can actually use any distance matrix. Doesn't have to be the phylogeny, but that's what we used. But we only use it for host gains. And we could potentially also add it so that it can affect the probability of host loss. Depending on the data set, because it's very rare that we have information about potential hosts, the convergence of the full model, the three-state model, can be tricky. It can be tricky to reach convergence, but that could be solved by adding what we call observational uncertainty. So basically saying that the states that we the states that we have at the tips of the data that we pass to the model, there's a probability attached to that. So the host, the actual hosts, are indeed actual hosts, because we've seen those interactions in nature. But for all the zeros in the matrix, not all of them are true zeros. Some of them are ones. So lots of the known hosts are, or at least some of the known hosts are potential hosts. And yeah, if we assign a probability to these states at the observed data, then we can maybe use the three-state model in more data sets. One tricky thing that we haven't figured out yet is how to calculate posterior probability of whole networks, or for like modules, so structures like that. And one other avenue that we are considering is to model this process outside of the tree. So simply have the process happen over time, and then at each time point you have extant parasite and host lineages, and then you have possible interactions. But that's sort of like future directions. And then very briefly, I just wanted to mention my current project, which sort of builds from all the work I talked about. So all the interactions that we observe in nature are part of the realized host repertoire. And this realized repertoire depends on two things: the available hosts for the parasite, so which hosts co-occur in space and time with the parasite, and also the fundamental host repertoire, which means which hosts the parasite is able to use. And if we want to predict interactions, we need to consider both these things. So this project, it's still in very early state, but the idea is to use an extension of this model of host repertoire evolution that I talked about to infer the fundamental host repertoire of extant European butterflies, then use species distribution modeling to infer the realized host repertoire for butterflies in the future according to different projections of climate and land use change. So which hosts will be available for which butterflies. And then combining these two, we will then be able to construct a probabilistic network for all butterflies and plants across Europe, hopefully. And based on that, we aim to quantify the stability, changes in stability and resilience of butterfly plant networks across climate change scenarios. All right. With that, I'd like to thank you for listening. Thank my former supervisors and postdoc hosts and longtime collaborators, Niklas Jans, Søren Elin, Fredrik Ronquist, and Michael Landis. Also thanks to funding and universities where I've worked. Thank you so much.
Wonderful. That was very, very interesting. So I invite people to post questions on YouTube. I'll just sort of start something off with like maybe just a bigger question. So you sort of addressed this at the very end, but I guess I'm just sort of curious, like, let's say we're able to run this analysis on 500 different host symbiont interactions. Are there sort of like some big evolutionary themes that we could pull out from an analysis like this? What would they be?
You mean in terms of theory?
Well, you talked in great detail. Yeah, in terms of evolutionary theory. I mean, you've talked in great detail about like a particular system, but if we were able to scale this up to like lots of things.
Yeah. So, well, first I can say that it's hard to say what are the limits of the method, but I have run analysis for like 1,300 butterfly species, including like all butterflies, but with a reduced number of hosts. So 16 hosts, I think I did, and that ran like in a couple of weeks and gave good results. So I'm confident about scaling it to larger data sets. So from other things that we have tried, so we have used it for a completely different trait, which is color patterns in mimicry rings. And that worked, and then we could, it's still not published, but so we used this model because then we could infer ancestral polymorphism. So that's one application that's possible. Then, well, there's been people that used it for other parasite systems, which are like questions more similar to mine. And then there's one that we're still gonna try that's for microbiomes in beetles. So, I mean, there's many different questions, but basically anything that includes evolution of species interactions.
Yeah, and that's sort of actually more where I was, that second part is more where I was going. So no, I wasn't thinking about like, can you scale it up in terms of like number of taxa for a particular analysis? But let's say you were able to clone yourself 500 times and each of your 500 clones was able to go out and like investigate a single system. And then after we gave a couple examples here, and then they all report back to you, and then you write some synthesis about all the things that you've learned. Like, I'm just asking more like, are there some bigger picture rules that we can infer from doing this flavor of analysis?
Yeah, absolutely. So I think I mean, one very sort of like fundamental question that I've always had is, what's the resource that species are after in other species? I mean, we use taxonomic groups as a proxy, but that, or phylogeny as a proxy, but that's of course not exactly what. Yeah, I mean, butterflies don't care about the name we give to the plants. So maybe as long as we have more data to incorporate or to combine with the analysis, then we could we could, for example, get more into the question of what is it that the parasites are going after. Is it more, I don't know, nutritious resources, or is it that they could use anything, but defense mechanisms don't allow them to interact with all of them? Or is it more geographic distribution that prunes or that constrains which interactions are possible? And then of course, like how general are these processes? Now I mean, it feels like we're sort of understanding how interactions between butterflies and plants have evolved, but how general is that? If like does that explain the patterns observed in other systems as well? So yeah,
that's cool. Yeah, I mean, I guess I don't know anything about these things, but like it's sort of, we're thinking about Chris Wheat's talk that was very, very sort of mechanistic and biochemical. Like it would be cool somehow to unite these two approaches where you make some hypotheses about host switches, but then maybe one could figure out something, how that actually came about.
Yeah, we've been talking, and I think I think we will be able to in the near future, at least in terms of chemistry and plant defense, get more into the actual traits that are driving these interactions, rather than just using the taxonomic names.
Yeah. Here's a question from Elizabeth S. Great talk, thanks. I was wondering how or to what extent plant adaptation is important to the rates in these models. Is plant adaptation slower than that of butterflies, and how does that impact things?
I'm not completely sure what you mean, but if you're talking about the plant changing over time, then I guess we can't really get at that, because of that assumption that we make that all the hosts must be older than the parasite lineages. So we don't really allow the host to evolve. That being said, you can, for example, look within the clade, so say brassicales. We could sort of reconstruct how different families within that order were colonized over time. And then, I mean, at least in that group, chemical defense really seems to be a major driver of those interactions, because those plants evolved more and more complex glucosinolates, and then the butterflies had to sort of evolve counter defenses to be able to use those plants. Yeah, I'm not sure if I answered the question. Please ask again if not.
I feel like that helps. I mean, the thing about the old host lineages, I think, like you say, is relevant here. So wait, okay, so the butterflies don't benefit from this. It's not a pollination sort of situation. It's sorry if I missed this.
So it's the caterpillars that
Oh,
feed
okay,
on
okay.
the plant.
Right.
Yeah,
Got it.
yeah. Then some butterflies also pollinize plants, but that's another set of interactions.
I mean, this isn't really a mechanistic thing, but it seems like something you could potentially go after, which is what is the form of this distance? Like you described that there's some multiplicative term that describes how easy it is to colonize something that's closely related. But could you add some parameters to that model to infer something about how good it is to be close, or what exactly it means to be close? I mean, I can imagine it being something where maybe if you're very close, then that's easy to colonize. But if you're above a certain threshold, then it's like doesn't matter at all, or something like that. Or maybe it's something that's sort of like continually, like as you get farther away, it gets progressively harder and harder to colonize. And I mean, who knows? But it seems like potentially possible for you to learn something about what that function looks like using a model like this.
Yeah. Well, in our function, I guess it's really just changing the equations, because we use an average phylogenetic distance from the new host to all the hosts in the current repertoire. But you could, for example, use only the distance to the closest host, or do calculate that in different ways. And then also the way that modifies the gain rate, that could also be different ways. In our case, we're only multiplying. So if the rate modifier is higher than 1, your rate is gonna increase. If it's lower than 1, your gain rate is gonna decrease. And yes, but that's just because we're simply multiplying. We could, if we change that equation, then we could do other shapes like you're saying. But then I guess we just need to like really have good reasons, like some hypothesis or hypothetical explanation for why it would have a given shape.
Yeah. I guess the hope would be that you would be able to just sort of infer directly from the data what sort of shapes made sense. But that's great. This is maybe not a it seemed the question of extinct lineages, I feel like, comes up. Are we not talking like
Yeah.
I mean, I don't know. I mean, it's you can see why I hesitated to ask the question, because it opens this huge can of worms. But I don't know. Even doing some simulations where you sort of do allow extinction and see what sort of effect it makes. This is totally a reviewer 3 comment. Sorry.
Yeah. No, but it's always a question that I get. We have no idea. We don't have extinction, and we don't know. But of course, that is, one thing that we did to sort of, because that question usually comes from the perspective of, okay, but there could be hosts that were used in the past that are not used anymore because they don't exist anymore. And then one thing that we did doesn't really solve that problem, but it's sort of a similar thing, is that we included more hosts than the butterflies use. So now the Pieridae that we include use, I think, 33 different plant families, and we included 50 families instead, so that we could cover the whole of angiosperms. And it's mostly like basal lineages that the butterflies don't use. But since we include them, they could have been used in the past. But at least in this case, because these butterflies are so conservative in their host repertoires, then we didn't reconstruct that. But it could be possible that they would have used a plant that in the past that it's not used in the present. But yeah, before extinction, I think, yeah, it would be really nice to do a simulation study just focused on that, and how that would affect also the structure of these ancestral networks. Yeah, so of course how the structure changes over time.
Cool. All right. Thanks for all those. Yeah, really interesting work, and I look forward to seeing how it develops.
Thanks. Thank you again for the invitation.
Yeah, thank you. All right, bye now.